get to work. All right. Are you gonna do this?
I can,
Or you go.
yeah, I can do it for you.
How can you see from there?
Oh, I can see.
Oh, you can.
All good.
Okay. All right, all good. Okay, so next slide, please. You sure you want me to move over here? Get close enough. Oh, I see, it's on now. Yeah. All right, so why study these? Okay, as I tried to allude to before, they're very common, right? The whole concept of evolution, right, is predicated on interaction between the environment and environmental effects and bottlenecks and so forth, operating on a sea of variants that are sitting out there, and then certain individuals in that population are better suited than others when environmental changes come. So those are critical elements to deciding why we have the genes and variants that we do have today because of those interactions, okay? Some of these things are kind of interesting and fun. I call these recreational observations, which I think you may have been exposed to, and I was bringing this up because a lot of students have been exposed to it. Some have told me that was their first sort of introduction to science and genetics and thought how interesting it was. And sometimes in your classroom in biology, you might have been asked by your teacher to taste different compounds and see if you could taste one. Did anybody do that anymore?
Yeah.
You did?
In high school, yeah.
And what happened?
I think it was like the strips. I had
Yes.
to taste, and some of them taste bitter, and some people others don't.
Yeah, and that's
Tastes bitter to me.
yeah, okay, and that's been mapped down to a variant in a single gene, right? So there's taste reception in one case, and then the more interesting one, the odor reception. I don't think they do that in school anymore, but so that gives an introduction to you as a student right away, right? That there's something in my genes that dictate my ability to taste or the bitter taste, as opposed to your colleague over there who, by no other reason would you think they're any different than you, but they're going, I can't taste a thing. I can't taste that difference. The same thing as when we taste food. People go, this doesn't taste right to me. You think, what's wrong with it? The food's fine. I just cooked it for you, My wife does this occasionally. Asparagus is one of those things that can taste different to people. And I go, I didn't do anything different, but again, that's the individuality you get with these kinds of traits. Why would it be important to have genes determine to some extent taste? Yeah, exactly. Right? You're all chewing down on stuff that's rotten and so forth. You ain't gonna last very long, or you're gonna have to have an iron stomach. And you see animals are able to tolerate certain things. Obviously, the way that they've adapted to tolerate certain things. But yes, you take poisons or other things, and you can learn from those behaviors also, right? If you can taste those things that may be maladaptive. Yep.
I was just going to say on the opposite end, like you might be attracted to things that have historically been very helpful. So if you're like, wow, this is a lot of fat, sugar in it, like I want to eat this. I'm not going to chew on grass because grass doesn't taste good to me. But
yes, but being a carnivore and getting a nice fatty meal and protein-laden meal is really going to be really good. And that's why particularly butter and things like that, right? Tastes great to you, well, most of us, because of the fat content, high caloric density, right? So we obviously genetically adapted, like a lot of other animals, to preferred diet as well as diet things you want to avoid. Okay? So it is critically important. And although I call that recreational, so just a nice example. Okay. There's this thing I keep bringing up. You guys are aware of genetic studies, right? Because you're trying to find the series of variants that contribute to a trait. The trait is the degree of heritability worked out by looking at populations, family studies. And yet we find when we do these studies, we only can explain a fraction of a percent or a few percent of the estimated heritability of a trait. So the thought is, what are we missing in these complex polygenic disorders? Maybe the interaction with the environment is more complex than we realize and is creating a situation where we've only got the genetic piece, and that's just explaining, as you can envision, a small fraction. Without the the environmental component, we can't work out some of the risks. So it's always been out there, and this was published 2012. We've really not made a lot of advances since these claims have been made by a number of the G1 papers, and we're missing heritability to be attributed to genetic environmental effects. So I'm still waiting for the first really major paper that's a solid example of filling that gap with the environment. I don't doubt it's likely to be possible, but we haven't yet been smart enough to really identify those environmental factors that are perhaps going to inform a GWAS association. So, one more, and he's ahead of me, thank you. There's a lot of medical implications. So, I wish my fellow physicians were using this information to a greater degree than they do nowadays. We'll talk about the examples, particularly with drugs. Next. So, you've seen this guy before, right? Garrett, and he really was, and the reason we bring them up, like myself and Dave and others that brought up Art Charles prior to them, is because this guy was very forward-thinking. They thought it was kind of crazy, actually, when he was at Oxford, but he came up with the idea of chemical individuality, right? That as he points out, idiosyncrasies in regard to drugs and articles of food, which are summed up in verse saying, well, if one man's meat is another man's poison, presumably have a chemical basis. We just discussed that. And we meant chemical, he meant inborn, because he hadn't been thinking about the genetics at the time dictated, so that's what he was getting at. Next slide. And he sort of, in a second paper he put out here, in inborn factors in disease, some effects vary from slight temporary discomfort to morbid syndrome amount of severe. So, over 100 years ago, he's getting at the idea of this individual response, which I glibly just explained to you. You guys go, oh yeah, we believe that, right? But when this guy came out with this, they thought it was crazy. I mean, he said, this is really outlandish, but like a lot of insightful individuals and their concepts, some of them really hold a lot of truth. Even evolution, still in some quarters as well in the United States, is not accepted, right, as a concept. But, we in general, even sometimes without direct experimental evidence, the circumstantial evidence is compelling. Same thing for here, right? The evidence is compelling that this is correct. We do have individual differences to the exposures. All right, next. All right, so here's now a single gene one, which is pretty clear and observed back in the 50s. That was even before my time, just to be honest with you. Has truly been informative and is used as one of the prime example of gene environment. You may have seen this before, right? So this is G6PD deficiency, X-linked disorder, and it's due to a defect in the dehydrogenase. Can't reach that far, so enzyme right here, which is important for this conversion, and the development of oxidizing things that can help reduce things and protect us against, reducing substances that help us against oxidation. And the oxidation causes problems. When you've got lots and lots of oxidation, your cells, your red cells, you get lots of red cells and hemolytic anemia, which can be fatal, okay? And there's a lot of people affected worldwide with this variant, okay, that carry this variant. And they're doing just fine. You wouldn't know they had it until they get exposed to something. It's really the males, and females are unfavorably lionized. Next slide. And what kind of things trigger this reaction? If you're a male or unfavorably lionized female, now you eat something or you consume a drug of some type that now puts additional oxidative stress on your system. Now you're not able in your red cells to compensate for this. And so sulfonamides, analgesics, aspirin, all these henna, flavins, beans. So red Mediterraneans have a diet heavy in beans, and those beans have a lot of oxidizing components. And I think it must be the fava beans that so if somebody has a nice bean meal, and all of a sudden they're feeling really sick and tired, and they're actually going through a hemolytic anemia. The red cells are bursting there, and it could be fatal, depending on how great. So people end up in emergency room with these kinds of episodes. Next. So, but otherwise, if they don't get exposed to those things, they're fine. And it might be, as we were saying earlier, you get exposed to beans, you go, gee, I felt terrible after I had those beans. My urine went all brown. The hemoglobin's now coming out of urine. And I got to touch those beans again, or so-and-so's cooking. If it's your mother's cooking, you better be very careful about what you say. So, but you will learn, if particularly in the second episode, or just do a little bit of this being, you get the same thing again, right? You can avoid it, right? So you can adapt to these things, but sometimes the first event is fatal. So anyway, but you can otherwise lead a normal health life. Doesn't affect things. Enzymes important in certain circumstances, but not in others. So we can pick this G6PD deficiency up easily, and we can also, by knowing about it ahead of time, if we do screening, or at least let families know, and families know that it's in the family, we could go ahead and inform people, say, be very careful with certain drugs, and follow these. Next. And henna, so forth. So, but why is G6PD so common? You guys know this, right? You do know this. You do know that this is also confers resistance to malaria, not just sickle cell, that variant, but also G6PD confers in your cells a resistance because the parasites really like to be in the wild type cells, not in the cells that have this G6PD deficiency. Okay, and so, as he says, the density of malaria infection correlates to incident mortality. Also, G6PD confers a survival advantage. Another survival advantage is another example of gene-environment interaction to consider. Well, next.
Oh, you know.
Okay. Oh, say next. Oh, sorry, I don't want to do it. So, and the males, it's interesting, have a degree of resistance is greater in general than the females, but there is some debate about this, interestingly enough, because you think, well, how's this getting passed on? Would you think it's the females would have resistance because it's right? They got two X's to pass it on. The male's not going to pass on the X except to his female offspring, not the male offspring. So this has been debated, and I tried to look this up last year. I didn't see any further information on this. But the interesting thing is, gene-environment interactions don't go one way. And what did you observe with the COVID pandemic? Which evolved faster, you or the virus? Darn right. It's a losing game. Okay? You've seen those sci-fi movies. We lose every time. Right? Those viruses out there, 12 monkeys, all those other ones, Viruses outwits us, and they're trying to. So, same thing here. Next. Plasmodium falciparum has found its way to make its own enzyme, G6PD. So, if you don't have it, I'll bring along with me. You can't select against me anymore. And guess what? Now that selecting advantage, that heterozygous advantage is gone. Okay? So, we have to be very careful what we do and how we modify things, because we don't fully understand relationships and we start thinking we're really smart about changing things. We can actually cause ourselves more damage than we realize. And in COVID, what happened? The spike protein mutated faster. What did it do, though? You guys who were living through this like me, what happened to the spike protein? What was the modification of the spike protein? Did it make the virus more lethal? It's a yes or no answer. No. No. No? Anybody else believe no, or is it yes? Let me give you an example of what you have to think about when you have to answer questions. You're working in a company and you're the lead scientist on this, and the suits come in to talk to you. And one of the suits, she asks you a question, like I just asked you. You gotta have an answer. Okay? Same thing if you're running a lab or you're out giving a presentation. Feel comfortable to give answers, even if you think you're not sure about it, or it might be wrong, because it's okay. All right, it's fine. I gotta get students used to saying what they think and realizing it's okay to be wrong in science, because we're wrong a lot more than we're right in science, right? So, where it's gonna go. But yes, it was lethal. Obviously, it was less lethal, guys. We're still here. If it was really lethal, it's like the sci-fi movies would all be gone. But the viruses want that. Actually, if you're selecting the viruses, I'm thinking about this, but what is beneficial if you were the virus thinking, what would really make me the winner of this race?
Ability to spread.
Ability to spread. I don't want to be lethal. I want to get reproduction in that individual so I can spread to other individuals and carry on. I don't know why they want to do this, but they do. That's what they're programmed to do. Yeah.
I remember also reading, lots of people talked about like the improvement or recovery from the HIV scare of like the 80s.
Yeah.
Lots of people were like, oh, we just got better treatments. But in many cases, wasn't it just because the HIV virus, many strains were less lethal than the original strains?
Yep. Perfect example. Thank you. Yep. So there is They adapt and we adapt. So we have to know which way we're going. And their adaptation is usually not to go to the post. So Ebola virus, which is really problematic, actually is nasty to get and can be lethal in many cases, but it doesn't spread that quickly, okay? Because you can't jump around. And the other thing you want is people to walk around and not know they're infected. Right? If you know they're infected, you isolate. So, consider these kind of things that the virus wants to do, or the bacteria wants to do, or something else wants to do in these interactions. They're complex. And they also, in the case of mammals or other animals, they evolve behaviors. All right, next. So here's some maps of the world and places where G6PD deficiency is common, and then the overlap with the malarial belt, and that's kind of the evidence epidemiologically that this is conferring resistance next. And here's one of those things I was saying. We're trying to do something positive, and we got to understand these relationships. So Lactat, which was a very effective drug against malaria, also was an oxidizing drug. So what did it do? Right. So now you start treating people. You've got populations where within these endemic populations, endemic malaria, you've now got a number of individuals who were selected over time through heterozygote advantage to be G6PD carriers, and then you give them Lactat, all of them, without testing them. Guess what happened? A whole bunch of people died from hemolytic anemia, and that drug, which is relatively expensive and highly effective for malaria, taken off the market. What could you have done if you're smart? You could have actually tested for G6PD deficiency ahead of time. Wouldn't have been that hard, actually. There are some field tests that can be done, point of care tests that can be done for this. So understand these relationships is essential to the way in which we want to interrupt these cycles, okay? That's why gene environment interaction is important. Next. Yeah, we'll jump. I'm not going to do that. All right, how do we define G by E? You look in the literature and you can sort of see a whole bunch of definitions. Genotype, environment coaction, different genotypes responding different to the same environment. Excellent. Some genotypes being more sensitive to changes in the environment than others, so different reaction ranges. Yep. Genetic influence on individual differences to susceptibility to particular environments. They're all sort of getting at the same thing, but I think they're slightly different flavors of the same concept. But I think they're all, from what I understand, as correct as they could be. It's just you have to be very careful of continuing looking at a gene by environment interaction. So let's go to the next slide. So the reason you got to be careful, and that's a bit what that American Journal review showed, is there's a variety of interaction occurring, and you've got to consider the models under which you think the interaction is occurring. Because it isn't always the same way, right? The interactions would differ. Here's an example, 3 genotypes, non-exposed risk. Then nothing happened, right? Only exposure, like G6PD, like you showed. Next. Here's another one. Actually, here's the genotype, and it does affect the risk. Here's the exposure, it flattens it. We have an example where we're looking at modifiers in CF. Steroid treatment, you cannot see the modifier's effect. And yet TCF7L2 is a modifier not only of diabetes and CF, but type 2 diabetes is the most prominent locus identified in GWAS. So he's got a p-value well above minus 110 or something like that now. Okay, so it's clearly an important locus, but treatments we do can eliminate that correlation. Next. And then you got ones that I think were also reviewed in that review, where they're going like this, right, in different directions. Okay. These ones you think should be the easiest to detect because you get incredible differences at the outlier population, right, of the AABB, the homozygotes, right, and the environmental effect when there is having a dramatic different risk for the individuals. The problem is, in a generally a stable population, that's 50% of your patients, right, or individuals. So you may or may not have enough to see this if you're not thinking of this kind of a model. But just keep in mind, it's not just going one way or the other, just like we talked about with taste. Taste could be advantageous in certain cases or deleterious, right? So you've got to think about the model. All right, next. This is another stuff. This is the boring stuff. Stuff I always hated as a student. But I've got to realize in science I think you really got to pay attention to the details. So it's really important you consider what the study design is, how they did the study. And all studies, as you know, have advantages and disadvantages. I'm not going to go through them all. I'll show you the slides, and if you want to memorize them, that's fine. Or if you want to refer back to them. But consider that each one has problems. It certainly has weaknesses, which is an important thing to consider. A lot of times what we use are cohort type studies. Sometimes case control. Case is not very good just because it's just such a mountain of other problems. Family studies are pretty good, but low power. And experimental, this one is really good. Your cells in a dish, your animals in an environment, your mice, your worms that you could work with. You really can do beautiful stuff. Humans don't like to be put, in a cage and exposed. They can't do the studies. But you can be astute sometimes and find exposures in humans that you could study. Okay? But these, I think, for us as scientists, we have to consider the importance of this and the ability to translate these kinds of observations because they are the most rigorous in my mind. And if you produce something in this realm and translate it to some of the studies that are done using these designs, even though they have disadvantages, I think you really can make advances. Next. What should be obvious to you guys, I remind myself too, you got to have the exposure. And the amount of people get exposed really dictate the power of your study, right? So as I'm like, if on the exposed individual sample, the frequency transfers the main effect of the genotype. Right? If you want to study risk factors underlying tuberculosis, which they've done, and everyone's been exposed. Everyone. You can't look at the interaction between the genes of the individuals and the environment choice. You've got to have sort of in the sweet spot somewhere between 20 and 80% of the individuals, or if you really want to rigorous, 30 to 70% of the individuals exposed because of the power of it, right? No one exposed, everyone exposed. Doesn't work. You got to get the opposite in the middle part, okay? It's not intuitively obvious when you first do these types of studies because there have been published studies where they had everyone exposed. Well, how can you tell when somebody doesn't get exposed? Where's your baseline? How do you make those kind of estimations in the lack of exposure? Next. Genotype frequency, pretty obvious to you, but it makes huge differences. Really rare variants, they're going to be difficult to study, and the real rare ones we have down here, you got to have massive numbers. That's why GWAS studies have 100,000 people, right? Because you got low effect of that variant, and you've got to study very large populations in order to achieve significance in a rigorous way. Next. And most important factor in my mind, reliability. Keep in mind, and don't when you work in the lab that you do a certain experiment. And the question you have to ask yourself, if I do that experiment again tomorrow, or next week, or next year, will I get the same answer? Right? It's got to be, if you're going to use something and make a difference, it's got to be a reliable measure, a reproducible. This is what the NIH and others are really getting on people's cases about, is reproducibility. Great, had a great paper in Nature. Unfortunately, Labs couldn't replicate your fantastic data. Guess what? How did you get that result? Were you objective, or was this just a one-off, or was it population difference, or something else? Well, you're rushing to be first when the two other papers in smaller journals like JVC and so forth, they've got results that can be reproduced, and they're different from yours. My thought would be, again, I'm philosophizing here, better be second and kind of more right than first and really wrong. So make sure your data is reproducible, and make sure when you read a paper, you ask, the measures they took in those humans or in that mouse, did they show you they could get the same result if they did that experiment again? How often did they reproduce that result, and how reliable is it? And a lot of human studies in the gene environment field have been not reproducible. And one of the reasons is because the measurement factors were crappy. Okay? So be rigorous and be critical of what they're using as the output. Next. All right, I'm going to go to another example here. and I've got to use this as a simplistic format, but one in which the field pretty much uses to evaluate gene-environment interaction. Okay, so it's a simple two by two table. Environmental exposure, no yes. Genetic factor, no yes. Simplistic one genetic factor. So here's your base. You set up one, right, for your risk. And now, if you have the environmental exposure, no genetic factor, that's your relative risk due to the environment. If you have the genetic factor on environmental exposure, that's your genetic. And now the two together, right? Next, please. Thank you very much. Sorry to be so. So, if this number, when you come out with it, is greater than the additive of these minus 1, well, you got an additive formula. But that's not gene-environment interaction. Next. You've really got to have this G by E be larger than the multiple of the two, right? Just adding the two together, well, you expect them to be additive, right? You expect one effect to add to the other. They're both in the same direction. That doesn't tell you the interaction between the two. That just tells you you've got an additive model. 1 plus 1 equals 2. Okay, great. That's not an interaction. Or 2 plus 2 equals 4. All right, and go to the next one. So let's give an example of applying this and how we think about it in a real world situation. Factor 5 lighting. All right, this is probably a clotting cascade, and this activated protein C, it cleaves the factor 5, and it reduces the activation, as I said here, thrombin clot formation. And Leiden is a variant in the factor 5 gene that interferes with the binding of protein C. So if you don't bind, next slide. This I want you to quickly memorize. And any MDs here? Turn this off, and I'm going to ask you a few questions. We all get to memorize this, unfortunately, and then you promptly forget it a year later after you're taking your exams over, because things have been added to this. But this is the key things that in medical school anyway, everybody ever memorized. And so this is the clotting cascade down to the actual fibrin, okay? And it's a beautiful system that's balanced by many other factors. It has to be, right? You don't want to bleed too much and you don't want to bleed too little. You've got to have a balanced system. And the defect we're talking about next is the problem with this protein C binding to this factor 5 Leiden when that variant is present. And this is relatively common in the population. So that's an issue, right? Now they're going to bleed. You're going to have a little predilection bleed. You don't bleed so much like with hemophilia such that it's a major problem, but you bleed in certain circumstances that it can be problematic. Okay? Next. Oh, and that's the actual genetic change that was found many years ago. Next. Okay. So a study came out in 1994. I told you I'd give you classic examples. And these are study sizes we used to get in those days, and we still do on occasion. Less than 100. Well, we got 100 controls there. But the point was clear. And it wasn't quite interpreted the correct way. So here's the factor 5 Leiden mutation present. And then they're looking at, and now we're talking about oral contraceptive use. And the problems with oral contraceptives, right, and whether or not these people had excessive bleeding, okay, while on oral contraceptive, which also can confer a shift in your coagulation profile, okay? So now you've got an environmental effect of coagulation and a genetic one. So what happens with the two? And should we know about this? So we are careful when we prescribe contraceptives and be aware of it, because there were some disastrous events that occurred with women on oral contraceptives. So this study was to start addressing this, and this was in this medical journal, Lancet. And so they took those that had factor 5 Leiden and didn't, and those with oral contraceptive use of none. And then they started working down the relative risk of having a significant bleeding event if you had the factor 5 Leiden mutation and versus no factor 5 Leiden when you had an oral contraceptive use. And he said, well, that 5 is greater than 3.7, and it's an important interaction, gene by environment interaction. But it's not. That's not right. Okay. Although it's published as such. Thank you for pushing that one. Actually, it isn't right, because let's go through the math of this, but their conclusion is correct, but the way you get to this is not quite correct. Next. Oh, there's a gap here. Yeah. So that's your baseline risk, right? Your no environmental effect or exposure and no genetic effect. Next. this is your environmental, these are the numbers which have environmental exposure, and your patients and controls are, patients being the individuals I had, unfortunately, events that were significant. Next, you've now got those that carry factor V light, this is the genetic effect, because that's in the absence of the exposure, and then finally, this group. So what they basically did is 25 times 100, 2 times 10, 20, and it's about, you divide it, you got the math I just quickly went through. And then you come up with number 5, that's something there, but that's not the way to do this. The way to do this is actually, and this is not me figuring this out, people who work on this all the time came out with a paper a year or two later, 2 groups, and say, wait a second, you got to recapitulate this. So I'm going over what they deduced, and again, this is recognized as the correct way to analyze it. But you really got to compare this distribution of risk between patients and controls, where the genetic effect and environmental effect are present against actually this baseline. Okay, so the way you got to derive it is the following: is the R by R for the environment is 8,400 by 2,200. So you do the math here. Okay, that's just basically taking 84 times that and that times that. Right? It's a simple kind of mathematical forms to do this calculus. So that's 3.7. That's correct. But now the genetic thing is a little different. Okay, it's not quite what they got, because it really should be this times that and this times divided by this times that. Okay. Okay. Sorry, I get pedantic with the math. I know you guys understand. But very important. It's really important. And it really surprised me when I read this many years ago. Because I was a junior faculty at the time, and was these numbers, because you go 25 times 100 and 2 times 36, you get what is really believed to be the gene-environment interaction, 34.7. So now it clearly has exceeded it in a multiplicative model, because there you apply those two, and now this exceeds it. So this is held up. This really is an interaction of importance. Yeah.
Question. I think I'm misunderstanding as I'm reading the experimental stuff. Who are the controls in factor V line mutation present?
Which one?
4, yeah, the 2 with oral contraceptive use and 4 without.
Yeah.
The controls are
Those are ones who didn't have the bleeding.
Yes.
the bleeding event. Okay.
Yeah. I know somebody understood this. Yeah, I mean, they're small numbers, so you can, come on, Gary, what's the actual power of this study? Yeah, okay, I agree. It's just an example for you, but you just have to trust that other studies have been done and supported this, okay? Then you have to ask your question, so what? Does it make any difference? Can you do anything? Knowing this, does it make any difference? Well, it certainly would have been laughed at if you considered what happened with that drug that was now not available for malaria. Malaria rates go way up when they take that drug out, and we could have identified those G6PD individuals ahead of time. So does it matter now? Next. So should you really screen people if they're going to have it? So what's the real risk here if they have this hereditary ABC resistance? Next. So now you have to consider some other things. Deep vein thrombosis itself, you have to consider the risk for that, because that's primarily the outcome that they were looking at in these, I said bleeding, I meant clotting, sorry. I haven't had my second cup of coffee yet today. I meant clotting events. So their deep vein thrombosis was the problem, not bleeding events. So, and of course, do you know what deep vein thrombosis can lead to? You do. Does anybody else? Yes. So clots break off, and where do they go?
Lungs.
Right, yeah, so you get clotted lungs and that kind of stuff, and it could be instantly lethal or very quickly lethal. Right? Pulmonary embolism. You're close, you have the pulmonary. And so, yeah, it's important, and that's what the cases were, and I apologize for saying lethal, I meant clotting. So, but that in young women is pretty common, okay? So, it's uncommon. So, is it a big deal that they get that healthy veins thing, not just like somebody my age, my veins are getting a little bit old, and I gotta get up when I get on a plane and sit around. Can't sit around too long, you gotta move around, because the risk factor for DVTs in older people goes up dramatically. It also goes up dramatically if you've had surgery, you've been lying around in the hospital, even if you're young. But if you're older and you're younger and you're active, you guys are fine. You have very low actual risk, okay? So now you have to think of, well, what is it, I consider the population risk. What does these factors think? Given the use of oral contraceptives is an important thing that you don't want to start changing because someone's got a factor-fied Leiden variant and take that way because there's risk for other things. So withholding oral contraceptives can, well, you get pregnant, and that can also bring on and or other kinds of issues. And so you can't just pull it away if you haven't really considered the real risk for the people who are primarily taking the medication. So next. So they decided, and I'll just cut, sorry, I have to hold on to this one. But anyway, so it was decided that actually when you calculate out the risk, you're not going to test for factor-fied Leiden if they're heterozygotes. Because when you calculate out the risk for them in the general population of having a DVT while on contraceptives, even with factor-fied Leiden, despite the fact that I showed there was an increased risk, it wasn't that relevant when you compare the importance of the availability of oral contraceptives, okay? And the consequences of not having oral contraceptives available to that person, okay? However, there are certain cases where that now changes, and that's what I'm showing here, the homozygotes. Homozygotes have much greater risk, as you can imagine, okay? And when you calculate out their risk, it's huge, 50 to 80 times those of no carriers. Thank you, again, through these quickly. And so now, if you've had a clotting event, you certainly did, as a young woman's clotting event twice, so off contraceptive is certainly on, you may very well get, or should get tested for factor V and a series of other coagulation factors to know your inherent risk. If you're a homozygote for factor V Leiden, I'm afraid you won't get to take oral contraceptives, okay? At least you'll have to have adjusted oral contraceptives to decrease clotting risk, okay? So, again, the information is held up over time, the gene-environment interaction is there, but the context in which you're going to apply this now has to be thoughtful. All right, next. And it's a relatively rare thing as applied there. Thanks. Yeah, and of course, if you had an event again, it's robust, I think I covered this. Thanks. Yeah, it's ahead of myself. All right, next. So, GYB examples, I'm going to give you a couple of them, sort of sampling this. In drugs, infection, and stress. See how much time we have to cover each of these. Let's go with the pharmacogenetics first. So, there are a lot of papers out there talking about interactions that people have with drugs. Right? Some respond really well to the drug, and some have horrendous results. Okay? And I don't know, have you guys been at Harvard yet and had Dick Roshan? All right, he will lecture on this. I think he's still doing his lecture. And this is a paper he covered this years ago summarizing. It's a beautiful paper. But Dick Roshan is up at Mayo, and in fact Mayo has actually put in place screening for variants in certain genes that confer different rates of metabolism of drugs. Drugs that we commonly give to people, okay, in the hospital. And what he and others have found over time is there's a whole series of variants in state. A lot of these liver-based cytochromes, others also in the liver and other places, muscles, and effects of polymorphisms with drugs that are commonly administered to people, like this one's paralytic, for example, or warfarin, right, to allow your blood to not clot when you have clotting predilection. And this is one of the classic ones. So this is why actually, I think it's Mayo has got a bunch of money because they came up with warfarin many, many years ago. And Dick figured out actually genetic variants that alter the effect of warfarin. So if somebody carries something that increases the effect of this drug that allows you to believe, you could believe more than the average person, more than the doctor will, you should believe, right? You're more than just preventing clotting, you're now causing more problems. So a lot of these drugs have either enhanced or decreased effect. These ones are almost all enhanced. Next. And you can distribute the population depending on the combinations you have, and if there's gene replication in certain loci. You could have people who like metabolizing certain drugs like nortriptyline, which is a psychoactive drug, okay? And they know when you put people on this drug, there's very different responses to this drug, okay? Some people do beautifully, some people do terribly, and other people have to come off the drug almost immediately because they're psychotic within a couple of days, okay? So, if you go ahead and take a look at these people, you can find people that are really rapid metabolizers, right? They sit at this end of the Gaussian distribution, and a few people out here. Imagine this is a real population distribution. There's going to be people way out on the side of the tail. And there's others, and there's this little group with these combinations of genotypes are poor metabolizers. What do doctors do? Do they tailor, when you get a drug from the doctor, does she or he tailor the drug to you? Do they? No, what do they do? They use a formula. Milligrams per kilogram, I'm going to do that. All of you in this room metabolize these drugs differently, right? All of them. It's all out there in the background of the population. We weren't selected on drug response. There was no environmental exposure to it until we came along with these chemicals and give it to people. So what happens? You're a poor metabolizer. Guess what the level of that drug in your blood is going to be after a few doses? Much higher as it builds up. You can't metabolize the drug. What about those who are on this end of the spectrum? Thank you. You're going to metabolize it quicker. Doctors say, well, maybe you don't respond to this drug. You know, maybe you're going to try something else. Right? Now, she or he may do a blood level on you and a test and see where you stabilize after a certain number of rounds, but they may not. Okay? A lot of times they don't. And I can tell you, I've talked to docs until I'm blue in the face about actually, why not check a genotype? Why not figure out what the CYP2D6 or other polymorphisms are in the individuals so you can sort of gauge how much you should give. It's not the way docs work all the time. Still, they give a dose, they see if there's response, and then they tailor the response. Why? Well, by the time I go and order the genetic test to figure out the polymorphism, I've got to treat the patient. I can't have the patient wait in my office for a week while the genetic lab goes and types. And even if it was relatively rapid, I can't wait 3 or 4 days. I've got to go do something today. That doesn't mean he can't do or she can't do some test later on, particularly if the patient's responding in an unexpected way. But it's just not part of medical practice yet. We're trying to change this, but we're not there yet. Anyway, next. Yeah, so there's the other direction. So the number of drugs for which variation is huge. Just about any drug you could talk about, there's variation in responses. So I pick out a whole series of ones here. I'm trying to remember, what's the next thing I'm trying to think? Yes, but I'm trying to make another observation here to point out that they put in this paper, is the frequency of poor metabolism phenotypes differs amongst populations. So the drug gets developed in, say, China, and they show fantastic effects of the drug across the population. There's some poor responders and good responders, but in general, the population responds really well. And then the drug, fine, we're going to now try it in another population, in European whites, or we're going to do it in some other population. And guess what? The drug doesn't work at all. Or in a large chunk of the population, it's poor in its effectiveness. And in fact, much of this can be explained by this variation in the background, okay, as you'd expect. Right? Background variation in which there is no selection, but it's still inherent there. All right, next. Tuberculosis is an unfortunate story. So isoniazid was extremely effective against tuberculosis, okay? And, as shown here, there is quite a few people that metabolize this drug rapidly, and others that kind of slow. It's almost as you can see, a bimodal distribution. And it's due to this NAT2 gene. And so, if you're going to treat isoniazid like a lot of bugs, you got to kill the bug effectively. Because if you don't kill the bug effectively, what will the bug do in you? Take a guess. Go on, you got it. What's going to do? What do these bugs do if you don't get the right level of killing efficacy to help your cells kill that bug off? What does the bug do? Well, yeah, stronger would be the term. It gets selected for the resistant strains. You're actually doing an experiment in your body. An environmental experiment. You're the environment, the bugs are in there, and you're underdosing that, and guess what? You're going to now select individuals who are carrying more resistant bugs. It's the whole basis of bacterial resistance, is incomplete treatment with some of these highly efficacious of these drugs. Penicillin, when it first came out, was so effective. Wiped out staph, strep, you name it. It's fantastic. And then they started using it by putting it in the water, like it was fluoride. everyone was getting treated with it. And guess what happened? Now you got resistant strains. Resistant strains not only to penicillin, but third generation cephalosporins have been developed, you name it. Even what's the one with V?
Vancomycin?
Thank you. Vancomycin. And that's really worrying because that's almost our last resort drug. And so we got to be aware of these kinds of interactions. We've got to be aware how we're going to be using these drugs, okay? So the problem here is resistant forms of TB occur because people who were fast accelerators were given sufficient doses. So now we have resistant forms of TB all over the world. And now you really have to treat these people for a very long period of time. But if you tested ahead of time, as Dick would say to you, and you knew that, you would have known what level of drug to give the patient, and you would have tailored it to the person instead of just saying, okay, it's X milligrams per kilogram average person. All right, next. All right, resistance to sepsis. I'm just going to put this one slide up. Why I love the beauty of models like this mouse model. You can see these different mouse strains from all the way from susceptible to certain infection, you might can't remember which of they were infected with, but that's not relevant, all the way to here, okay? You could probably stratify humans to this, but again, you can't infect a bunch of you guys in the audience and you're sick with, how bad you go and how colonies you form. So, but mice, you can do this, and we know for sure that strain differences, there's got to be genetic differences, right? Strains are different genetically. So clearly we know this thing. And there are other examples that we have in humans. Next. HIV and the CCR5 delta 32. Do you know this story? No? Yes? No? Well, I'm going to cover it quickly anyway. So CCR5, right, is a chemokine receptor, and it facilitates HIV binding. This is like junior high school kind of graphics here, but it works. But anyway, it binds here, and there's co-receptors of which CCR5 is important in the binding and internalization of viruses, how the virus infects us, okay? Part of that. And as found by some colleagues out in Fredericksburg, I think it was, there was 32 amino acid deletion. That was relatively common in the white population, actually, white European-derived population. And it confers resistance because the co-receptor missing that 32 amino acids, HIV can't bind it. It won't bind efficiently, so it can't internalize it. So you have a certain degree of resistance conferred to you against something that was, as far as we know, not affected in the human population 100 years ago, but it's out there, it's an entity. And the homozygotes are almost completely immune to it. Completely immune. And discovering this was really important because it led to us thinking about what kind of drugs we need to develop in order to combat HIV, which has been a success story. Next.
Do you think that if targeting like CCR5, which I think has been people's interest, do you think that the viruses may able to evolve to bind new receptors, or how likely is that?
I'm going to guess you're correct in that thesis, and I don't have an answer for you. But I would suspect, and that's the problem with the viruses, is they may adapt to use others, because it isn't just that co-receptor or other place in the body. But I don't have an answer for a specific, say, yes, I know, and here's the paper to say. But I would suspect that it can. And that's the issue, is that, as I said before, the viruses can out-evolve us, and why they're so dangerous.
Are there like any CCR5, like binding small molecules that exist?
Yeah, well, they try to target this, right? That's part of the targeting as well as the viral reproduction as well. So this is part of sort of understanding how it does it. So finding a genetic variant that confers resistance to environmental exposure, which you have to have exposure, right? Now you got a underlying genetic effect that has, so homozygotes have no phenotype difference, right? From those who are hets, you wouldn't know. Like somebody in this room could be a heterozygote, for example, might be a homozygote. You wouldn't know it until you get infected, okay? Or exposed. Exposed, not infected, exposed. So that's the other thing, you've got to establish exposure to know this. And really what came out of the heterozygotes, yeah. But the problem is, as you're pointing out, once you start then monopolizing that, putting a drug in that thing, well, the virus is able to, because it trillions of them each time there's infection, right? What you need is a slip up or a shift in the replication of some gene or something within the viral genome, and it might now confer the ability of that virus to infect. If it's a marginal change, guys, right, a little tiny change can make a huge difference in something that has a very short generation time. Okay. So, but that was an important discovery, and it helps develop effective therapies for HIV next. And so, why is it there? Oh, 14% I was exaggerating the numbers. Why is it there, and why on other populations? Is it something to do with other infections that occur in the population? Now we get into the role of wild speculation. Right? I mean, Black Death, that, people are trying it, and they do experimental things, bronchiation. You know, until we get a time machine, and we can go properly sample, we'd have no idea what happened. But there's been a lot of ideas out there, and then, so maybe it conferred in some prior time some advantage, or maybe not. Maybe it was just sitting there all that time just by chance. And it was some kind of genetic drift that got the frequency up to about 14%. Wait a minute. Next. And then people came and said, well, wait a second, we have some experimental results, so it goes the other way. Maybe there's a problem with things like smallpox. Maybe it plays a role in a different way. So next, sorry, it's resistance to smallpox, but then people did studies with the West Nile virus. That's it. I forgot that part. So they were saying, well, maybe bubonic plague, maybe smallpox, rampant in places like Europe. But the problem is, you do know that those infections were rampant in other places in the world, right? China, and Africa, and every place else. So why there? Well, there's no good epidemiologic argument here. West Nile, you could sort of think it's more regional, but they are claiming in certain cases maybe those carriers are prone to it. So it's not settled yet, but I would suspect it's got nothing to do with anything else. It's just there in the background, and it just happened to get discovered. Maybe it's HIV throughout the population. Next. COVID. We talked a little bit about COVID. I don't know if you followed the story, but trying to understand risk factors or genetic risk factors to alter your response, because some people get the COVID-2 virus and do just fine, and others have, unfortunately, life-threatening events, and work done by a large consortium for a series of genes they found by a GWAS study that all fell into this interferon pathway. And then, next slide, and then this paper in Nature, very interesting, provided some biochemical support of the role of this pathway in infection with COVID-19, and I thought it was brilliant to study this, and I didn't pull the paper next. I'm going to jump off that because another paper, a couple other papers came out, did GWAS, and couldn't replicate the findings. So, okay, that's the importance of replication, right? You had a really good story, maybe all these interferon genes play a role, and of course you know the interferon genes play a role because they're part of the innate immunity, right? So you know they got to be playing a role, so you start mucking around with them. The question is whether those variants in particular are contributing resistance to COVID. And I guess it still needs to be further tested. There's probably some truth in all of this, but it couldn't be easily replicated when they use larger population studies. G by E and depression. So many of the gene environment studies you'll see, and I think was alluded to in the years I gave you, have been done in the psychiatric fields. Okay, we're looking at why do some people under certain stresses respond one way, and why do they respond another way? What's the background of it? How is it we as humans, when faced with certain lifetime stresses, some humans develop a severe and horrible depression in response to those stresses, and others will appear to be on the same stresses or the same family environment. If it's abusive, for example, they don't. And is it genetically possible? Possible due to genes, right? Not reasonable hypothesis. So years ago, this is back in the 70s, I think originally, people started figuring out several pathways that were important for healthy mentation and mood, and these serotonin receptor genes, transporter genes, were cloned and identified, and then a variant was found, a lens variant, in this particular one, and it's encoded by this SLC6A4, and people started studying this next slide and saying, okay, if we see a short and a long version of this, how does it, the long version associated with high serotonin uptake and the short version to low serotonin uptake. And serotonin is really important in mood stabilization. Okay, so could it be that depending on whether or not you're efficiently uptake the serotonin or don't efficiently uptake, that when you get stressful events, environment, and here's the genetic variant, right, the genotype, does it change this outcome, probability of episodes of depression? Okay, this paper by Athg was published in Science, and people went, wow. I mean, it really was a remarkable finding, and it stood up statistically quite robust, and look, 0.4 plus a very big difference. So why is that kind of observation so important?
Same environment, different genetics.
Yeah. What has the genetics told you guys? Any place you want to make a drug is going to make a lot of money, help a lot of people. Maybe the serotonin system, and maybe the uptake system. You do notice a whole large class of drugs based on that, right? This is part of the reason they did it, okay? And those drugs do work. They are effective. Not in everyone, but they work. So these kind of clues are important when you find them, okay? Because sometimes Mother Nature, by the fact that there's an interaction between the gene or genetic variants that we have and environment, gives us clues as to how we could treat people, for example, who have these kind of horrible events in their life. Okay. But I don't want to simplify it. Depression, schizophrenia, a lot of psychiatric are very complex disorders. This is simplifying greatly what's done here, okay? Because this has been contested a lot, although I think the concept is held up. Next slide, please. And there's been a revision of this model. It's not a pure monogenic system. It's not just good uptake, you're more resistant to this, poor uptake, you're more likely to get stressed, because they find, nah, it don't really hold. The idea is it's a plasticity thing, and if you're in a more positive environment, so there really is an interaction, but it's much more complex, then you have decreased symptoms versus a negative environment. So it's more complex about the environment. It's not just stressors and other things like that, okay? And it's the ability of the individual to how they do in those environments. So it's an interaction between your genome and the environment you're in, not just, okay, I carry that variant, it doesn't uptake serotonin that way, I'm going to be more likely to get depression. Because there's a bunch of people carrying that same genetic variants, a lot of stresses, and they were fine, never had depression. So you've got to explain that. Why is that? So it's, again, a susceptibility issue. All right, next. And I'm skipping through a few things. Again, I'll give you the slides because we started late and I don't want to hold you late. My stomach's going to start rumbling for lunch. Yeah, but you'll get through it quicker for me. Thank you. I do want to just cover this at the end. Gene-environment interaction, right? The multiple interaction between the gene's environment versus correlation. Kind of an important thing because genetic influence on individual difference in susceptibility to particular environments, but this is genetic influence on individual genes, exposure. You know, maybe you've seen the papers on variants that have been associated with risk-taking. Right? I don't know if you've seen these, right? The more likely to jump out of airplanes, or more likely to do, must be also male-related, I assume, if they're jumping or driving cars fast, or getting accidents. So, you know, you can say, well, you know, if the variants themselves modify the behavior, people may put themselves in environments that are more risky. And so, for example, if you got a variant that influences your anxiety and your response to stress, you may, because of that, put yourself, unfortunately, in more stressful environments and therefore get worse outcomes. So that's not really a gene-environment interaction. It's basically the genetics driving your behavior or your environmental exposure. Okay? So
How do I think, I guess, your philosophy, I guess, being an expert in this, how do other experts in the field kind of stray away from how much weight they give to like variant associated, like you use the one example, like a risk-taking variant, for example. Well, how do you deal with caveat with saying like, oh, variants can be associated with behavior? How do you ensure that that statement can't be misused.
It certainly can be. Point's well taken, right? Because it depends on how you sample, right? Well, I find more people doing listening with that variant. Well, it could be, there's lots of reasons that could be occurring. That's why replication is important in the house. But I think there's assumptions about variance and behavior, right, are based on many observations and replicated in many different situations, right? So if that variance has been associated with holistic behavior, then you might find higher, not only people jumping out of airplanes or driving cars fast to get in car accidents, you might also see they're also more likely to, jet ski or to go snowboarding. So you have to see it in various environments, and this is what they've claimed. I still think I'm not saying I'm an expert. I've read the literature, I'm not so sure myself. It's an amusing concept, and it's not unreasonable to think that we may have genetic variants that alter our behavior. I mean, we know people that want to jump out of airplanes. I don't. Maybe it's my genetics I can blame. Or maybe something else, but so what I'm trying to get across here is studies that look at gene-environment interaction or correlation in certain cases, because we are complex individuals, particularly in the behavioral realm and in humans, it's very complex. Be very careful and critical of observations you see regarding these things, because humans can adapt on the fly too, and their cognition and approach to things very fast, right? And so trying to interpret these relationships consistently is difficult. That's why many studies have to be done for us to be advanced.
The idea directly contradicts like the exposure theory and like phantom limb that you want to decrease the more times you have that stimulus.
Phantom limb, so you're saying the perception is still there after you've had an amputation, for example?
And if different genetic factors are pushing people to have more adrenaline-seeking events, there's also the counter idea that having more exposure to your phantom limb, or just an acceptance of your phantom limb, decreases that pain.
I don't think those are necessarily contrary, because you're still
I'm
trying
trying to answer it, to
to,
be honest, because I don't know enough about the phantom limb, so I think, yeah, go ahead.
you're still trying to influence your exposures in order to change your reaction, right? The opposite direction.
Yeah, go ahead a few slides and we can talk about this. Yeah, so let's talk a little bit about this. I'm not going to do the Roslin example, it's with it. But this one here was interesting. So this came out as a paper. It's a classic case of gene-environment interaction. Okay? So this, I think, brings up a little bit of discussion down here, if you think about this. Next. So there's a variant associated with nicotine dependence, right? So it varies your dependence thing. So what they found in this study, they got this genotype, and it alters nicotine dependence and so forth. Hang on. I'll do it one time. Please, I sort of remember what's in here. So this is in a drug that metabolizes nicotine, okay, acetylcholine, or responds to nicotine, acetylcholine receptor. And so that genotype alters your response. You smoke cigarettes and gives you a bit more of a buzz, right? But what are you going to do with that buzz? Your smoking quantity could be influenced, right, by the buzz you get, because that's your behavior, is how many cigarettes you get. And so if it gives you a really great buzz and you love it, and you smoke more, what might happen? Next. Your rate of lung cancer may go up because you smoke, and you smoke a lot more, because lung cancer rates are directly related, in the smoking case, for how much you actually smoked. Okay? So their claim was, this genotype also influences lung cancer. Right? Smoking, genotype, cancer. So it's a gene-environment interaction. But actually, maybe it's influencing through the nicotine dependence and the feedback you get, you smoke more. So next. And you also get peripheral arteries. So it could be that nicotine dependence is working on smoking quantity. And then what they didn't mention in the paper, but has since been revisited and has been advised now, next, is the fact that smoking quantity is there. That relationship had to be recognized. And so is it really a gene-environment interaction, or is it the genotype driving behavior? So is it a correlation between gene and environment? Okay. Is there another arrow? Yeah, provider. So I'll leave you with that. That's my 2 points there. And I won't go through, but if you look this up, this example up, you will see people who have written on this question of the G by E versus the correlation I mentioned. So even for the really good people, these are the real experts, it's difficult to tease this apart because if you hadn't fully considered some of the other factors, I did a simplistic, but this was shown through mathematical elements and modeling that really the big driver was the smoking quantity of cancer, not the negative, not this relationship is what they're claiming. All right, last but not least, I think. And for your reading pleasure, there's a whole series of really good studies out there. I think this is a beautiful review in American Journal of Epidemiology that really did outline. Many of them are drug interactions, but there are other ones like traffic pollution for respiratory with this nitrous oxide and others. These studies really were really good. I mean, really felt to be passing the test that I've just put to you. Properly powered, appropriate outcomes, define the best way. So there are studies that have been successful out there. I just showed you a list and another thing. But if you wanted to read more about some of these examples, I'll leave it. They'll be in the slides if you want to take a look, or you've got a particular interest in a certain area of interactions. This is a good list. And if you look through the literature, you'll find other summaries frequently done by the epidemiologists that vet these and show these ones really do seem to pass the test with A, B, C, and D. So please be critical when you see this, but recognize really it's there. But teasing it out and being sure you've got relationships correctly is the tricky part. And that's why you have to do some modeling ahead of time. Once you have the model in hand, then go and test. And accept if it doesn't fit your model. Maybe it's not really there. But you've got to get the model first. And that's why I gave you that review. Okay? Thanks for your attention. Sorry I started late, a little late. And thanks for doing the slides. All right.
I was going to ask,
Yeah.
is it possible to ensure that really only clinicians get access to this kind of data? For example, you talked about a risk allele that was associated with nicotine dependence. And so ensuring that in these kind of studies, the conclusions drawn, I guess if it's for a patient going into a clinic, right, and we're in a world where genetics is super in the clinic, they see you have a variant and they just know to monitor patient smoking usage or whatever. Because it might not, like you said, without the exposure, they might not ever know they might have this variant.
Yeah. I try to bring that challenge up with the fact of life. I mean, yes, we know it, the association. We've got to think then, how do we use the information effectively? So, knowing you have a variant that changes your nicotine acetylcholine receptor and makes you more susceptible to the positive effects of smoking, how many other factors might be involved and so forth? And so, the problem in a lot of genetics right now, in the complexes or with many genes and loci involved, the effect size is small. Right? There's so many other factors, including environmental factors and gene environmental factors. Monogenic disorders, where you got a high predict, that's a different story, right? So for most cases, but a lot of these other ones, these relationships are all based on the interaction of multiple genes with multiple environmental effects, which is why I started out telling you it becomes incredibly difficult from a calculation point of view. But I don't think it's going to be impossible because in your generation, in your careers, you're going to be able to deal with masses of data. And you're going to have much larger data sets because electronic patient records, genome data, many of us will be genome sequenced. You guys can do this. You guys will be able to sit down and figure this out on the computer, some of these interactions. Things we couldn't do before. You'll also use machine type learning, which is what most of AI is. We'll take it and see patterns that we don't see, that we can't even predict. So I think you will figure out a bunch of these things. It's a really interesting area to go into and very important for our health and disease and everything else.
Would you say the environment then in this case is probably the most significant limiting factor? Considering these studies, because I guess you could probably say every single human being has some variant or variants that phenotype-wise only show based on whatever environment. So
Let's see if I'm getting at what you think of.
Yeah.
We all have the variants, but the point is, what tools do we have to go after variants? Really effective, right? Sequence a genome now.
Yeah.
You also can probably get methylation status. Soon you'll be able to get chromatin status and everything else and the activity, and soon we'll have a better idea about enhancers and gene regulation. That part in the sequencing is almost easy. But as you're pointing out, getting the environmental factors, I don't even remember what I had for breakfast this morning, right? But if it was something that we need to know about, so that we think, we have to collect that data on humans in a very dense way. When you do these studies of mice, you can collect all the data you want on that mouse, and that's why you have a much better a way to phenotype them actually and their exposures. Can it be done in humans? Well, we have to decide if we want to be, put in a box and track and all that kind of stuff. I don't mean a real box, but be willing to document our exposures and those kinds of things. But there are studies of people who live closer to a highway versus further away. And we know particulate exposure can be worse. And now we've got more and more good evidence showing that particulate exposure is directly related to rates of lung cancer. Okay? Become a major risk factor. We're now with some epidemiologic studies. And there are individuals within those groups with certain genetic variants that have even greater exposure. And these are germ lines passed down, right? So it's a new world. We took the easy, low hanging fruit, my generation, on genetic disorders. You guys get the hard stuff, but you have great tools. And if you get interested in doing this, I think you can have some really interesting observations that will make a big difference for a lot of you. All right? So thanks for your attention. Any further questions or anything else, shoot them to me, or something I said wasn't clear as I barreled through this talk. Let me know. I'll send them. Who's responsible for the slides? I'm going to send to you. You guys?
I already have all the slides.
You have all the slides already? Did I send it to you?
They're on Canvas.
You're on Canvas from last year.
Great. Oh, from last year I didn't update. Okay, yeah. You guys can catch me easily, right? Yeah, you got it. And if there's questions about the slides or anything else, let me know. All right. Thank you very much.
Thank you.